get it eventually.
Okay, can everyone see this my screen?
Yes.
Right. Okay. Okay, so welcome back to those of you who are returning, and welcome for those of you who are new here. So today's session 2 of Molecular Biology 101. So we're going to be focusing on DNA today. So for the first half of this session, we're going to focus on more conceptual topics. So that includes higher order DNA structure and packaging, DNA replication, and DNA damage and repair mechanisms. Then we're going to take a short break and then come back for any questions you have. And we're going to do a really short question set. And then the second half of the session will be we'll focus on DNA-centric techniques in the lab. So we're going to talk about DNA purification, gel electrophoresis, PCR, restriction enzymes, and molecular cloning. Then we'll take another short break and then come back for a few more question sets. So part one, we're going to start with DNA structure and organization. So this is where we left off in the first session. So what we went over in the last session was that DNA is in the structure of a double helix that consists of a phosphor sugar backbone that's on the outside here, as well as complementary nitrogenous bases, which are represented in the middle here. In addition, the two strands are in an anti-parallel conformation. So that's the simplest level of DNA structure. So the double strand helix of DNA is wrapped around proteins known as histones. So histones are rich in lysine and arginine amino acids. So some of you may remember from last week, arginine and lysine are both positively charged amino acids. DNA is negatively charged, so the difference in charge contributes to the complex formation between DNA and histones. So there are 5 types of histones, 4 of which comprise the core of a nucleosome. So nucleosome consists of 8 histone molecules. They're called H2A, H2B, H3, and H4. And each nucleosome, the DNA wraps around each nucleosome 1.65 times. And you can see the structure here in an electron micrograph. These dots represent the histone cores. So the nucleosomes are attached by a linker histone, which is a histone H1, and this forms a chromatosom. The chromatosomes are then folded to produce a fiber that's approximately 30 nanometers. And this forms loops that average about 300 nanometers in length, which are then compressed and folded to produce a fiber that's approximately 250 nanometers wide. And coiling of this fiber results in the chromatid structure that we're familiar with. So the form of chromosome that's represented here is actually consists of 2 sister chromatids. So in diploid cells, which are cells that contain 2 copies of DNA, one from each parent, they have what's called homologous chromosomes. So those are chromosomes that are that are equivalent, that one comes from each parent. During the cell cycle, these chromosomes replicate to create sister chromatids. So these are identical copies of DNA, which are then divided when the cell divides. During DNA replication, the number of chromosomes remains the same, but the number of chromatids doubles. So that's represented here. In addition, the larger structure of chromosomes also contains this structure called the centromere that's located at the center of the chromosome. This is where the spindle attaches during cell division. So there are some differences between prokaryotic and eukaryotic DNA structure. So what are some differences between prokaryotes and eukaryotes that we're already familiar with? Feel free to just jump in and list any differences you already know of. Any volunteers?
What is more about?
Well, there's.
I think I heard Pedro start to say something. Please go. Or Kiki, I see you've unmuted.
Yeah, I just heard someone else was talking, so I didn't know if I should go or not. So prokaryotic DNA is usually like bacterial DNA is it doesn't have an actual shape, like it doesn't form chromosomes, it's just circular. And prokaryotic cells are like they usually have a cell wall, and I do know that eukaryotic for like fungi and plants do have a cell wall, but otherwise it's just the cell membrane. I'm sorry. And also prokaryotic cells do have plasmids, and that way to like share information in between each other, but eukaryotic cells cannot do that, as far as I know.
Great. Anyone else have anything to add?
Are prokaryotes, don't they, is the nucleus enclosed? Isn't it like just in the whole cell? Does it have a membrane around it?
Prokaryotes, they don't have a membrane-bound nucleus. They have a region of the cytoplasm that's somewhat equivalent to a nucleus, but it's not membrane-bound. Anyone else? Okay, so this lists out some of the major differences. Yeah, like Kiki mentioned, most prokaryotes contain a single chromosome that's in a circular form. They're condensed in a region of the cytoplasm known as a nucleoid, as opposed to in eukaryotes where there's a defined nucleus with a membrane. In addition, prokaryotic DNA can undergo transcription and translation at the same time because it's located in the cytoplasm. They're typically haploid, which means they only contain one copy of each gene. And non-essential genes are encoded on extrachromosomal structures known as plasmids, which we'll be talking about later in the context of how plasmids can be manipulated in a laboratory setting. Also, prokaryotic genomes are typically more compact. They contain less repetitive DNA compared to eukaryotic chromosomes. So this is the prokaryotic DNA structure. It's a circular chromosome, and then it is looped using actual proteins to create these loop domains, and then it's supercoiled. So, and here is an electron graph showing how
Can you
that
say what you
actually
mean by
looks.
supercoiled?
It's just compressed further. Okay, any questions about DNA structure?
Do prokaryotes have histones? Is that how they supercoil, or do they have something else?
They have another protein, but it's not. They're not histones.
So they do get wound around some kind of protein. They're not just.
The loop domains are formed by those proteins. The supercoiling, I think, is partly due to topoisomerases, which we'll talk more about in the replications in a second.
Oh, okay.
Okay, so we're going to get into DNA replication now. So DNA replication begins at a specific sequence in the DNA called an origin. There's a number of proteins referred to as the origin recognition complex that are recruited to the origin of DNA, origin of DNA replication, and they basically open the double helix, and that allows access for the replication machinery to load onto the DNA. So replication requires an enzyme to read the template that also has a catalytic function to form the phosphodiester bonds. DNA replication occurs in the 5' to 3' direction. The enzyme that carries this out is called DNA polymerase. It loads onto one strand, but it reads the bases from the opposite strand, and then it selects the proper bases to put onto the new strand, and then it joins the new base to the new strand through a phosphodiester bond. In addition, there's an exonuclease that's present for proofreading function. So this reads from the 3' to 5' direction, and it reads the base pair that was just created to check if it's correct. If it isn't, it removes the base pair. And this is the catalytic mechanism of DNA polymerase. So it connects the 3' hydroxyl group to the innermost phosphate group at the 5' end of the following base, releasing two phosphate groups. This is a schematic of the DNA replication fork. So as I mentioned before, when the DNA replication proteins are loaded onto the origin of replication, it needs to open up the double strand of DNA so that the machinery can access the single strands. So some of the proteins that are involved in this process include the DNA polymerase, which I just mentioned. DNA polymerase cannot start synthesizing DNA de novo or from nothing. So it needs a short sequence to act as a primer that it can add more nucleotides to. The enzyme that creates this primer is called primase. Usually it's an RNA polymerase. It adds a short sequence of RNA to which the DNA polymerase adds new base pairs to. This process also causes single-stranded DNA to be exposed. Single-stranded nucleic acids are not very stable in the cell, so they need a protein to bind to them to keep them open and also protect them from exonucleases or enzymes that will cut single-stranded DNA. So that enzyme is called the single-strand binding protein. Here, we're just going to refer to it as SSB. Next is helicase, which is represented here. So helicases function to separate the two DNA strands. This will create tension, which then needs to be relieved. So the enzyme that deals with the tension is called topoisomerase, and that controls the twisting of the DNA so that it is able to separate. Lastly, there is the RNA primer needs to be removed. So there's an enzyme involved in removing the RNA primer, and then there's also DNA ligase involved in this process. The reason why there's DNA ligase involved is because the two strands of new DNA are being synthesized in different manners. So you have what's called a leading strand and a lagging strand. Because DNA polymerase can only work from the 5' to 3' direction, the leading strand can be synthesized in one continuous action. The lagging strand, however, is in the opposite orientation, so it can only form short sequences with new primers being added at intervals. So it creates a series of fragmented DNA referred to as Okazaki fragments. You can see here. So once DNA replication is completed, DNA ligase has to anneal the Okazaki fragments together to create a continuous strand of DNA on the lagging strand. And this is just a close-up of the difference between the leading and lagging strand. So here you have the leading strand, which starts at the 5' end here, and it continues towards the 3' end with no breaks. The lagging strand, however, has to have a short RNA primer, and then it creates an Okazaki fragment in the 3' direction, and then a few base pairs down, it repeats this process. Then these RNA primers are then removed, and you have a continuous strand at the leading strand, but you have short Okazaki fragments at the lagging strand. These gaps are then filled by DNA polymerase, and then these segments are sealed or annealed together by DNA ligase. And we're going to go over some differences between prokaryotic and eukaryotic replication. As we've mentioned before, prokaryotes don't have a membrane-bound nucleus. So DNA replication occurs in the cytoplasm. Because there's only one chromosome, there's only one origin of replication per molecule of DNA. Eukaryotic DNA is it's more complex, so there are multiple origins of replication within each chromosome. In addition, origin of replication is usually 100 to 200 or more nucleotides in length, whereas in eukaryotes, it's approximately 150 nucleotides. Because prokaryotes only have one origin of replication only occurs at one point in each chromosome, whereas in eukaryotes, because there are multiple origins, it can occur at several points simultaneously within each chromosome. In addition, the actual enzymes involved in both are different, but that is a little too much detail for the scope of this class, so I won't be going into that. Are there any questions about DNA replication?
I don't know, there's topoisomerase or topoisomerase. Those are really cool because when you think about the DNA strand, since it's all wrapped around, when the replication fork, when the helicase is opening it up, as you pull it apart, it's like pulling two strings that are wound together. You pull it apart, and then it's going to knot up downstream. And then those topoisomerases come along and they cut one strand and pass it underneath to unwind it. So it's like a little person with a string, like cutting and moving it to the other side and tying it up again and then cutting again. And it's like racing ahead of the whole replication for it to keep the process going. It's just kind of mind-bogglingly intricate and amazing that just wanted to share that. It's one of my enthusiasms about the subject.
Okay, if there's no questions, we can move on to DNA damage and repair. So I'm sure we're all familiar with forms of DNA damage. Can anyone there are some sources of DNA damage out?
Sunburns, my least favorite.
Yeah, other types of radiation.
Anyone else? Okay. Yeah. So UV and radiation are both examples of DNA damaging agents. DNA damaging agents fall into one of two categories. Either they're exogenous or they're endogenous. The main form of endogenous DNA damage comes from DNA replication errors. So if there's a defect in the proofreading function of an exonucleus, that can cause DNA mismatches, which is a form of DNA damage. Exogenous forms of DNA damage or DNA damage agents can include X-rays, oxygen radicals, alkylating agents, random spontaneous reactions, as well as UV light, aromatic hydrocarbons, X-rays, and anti-tumor agents. So these cause specific types of DNA damage. So for instance, X-rays and reactive oxidating species and alkylating agents can result in alterations to the nitrogenous bases. So this includes like things such as diamine being converted to uracil, the loss of nitrogenous base, which results in an a basic site, or alterations in guanine, which results in this 8-oxoguanine variant, as well as single-strand breaks. UV light exposure and aromatic hydrocarbons can result in the formation of dimers between purines or pyrimidines. So this is what a 6,4 photo product is. It's a dimer between two adjacent purine bases. This can polycyclic aromatic hydrocarbons can result in bulky adducts, where a region of DNA is bound to toxic chemical. And then X-rays and anti-tumor agents can result in double-strand breaks or in intra-strand crosslinks, which are shown here. It's crosslinks between within one strand, between two strands, through a guanine binding. And replication errors can result in mismatches, insertions, and deletions. So for each of these types of DNA damage, there's a specific repair pathway that can resolve it. So so these include the base excision repair pathway, which is used when a nitrogenous base is damaged or missing. There's nucleotide excision repair, which is used when a nucleotide is damaged or altered in any way, or if there's something bound to the DNA. And then in the case of double-strand breaks or interstrand crosslinks, there are two pathways that can be used, either homologous recombination or non-homologous end joining. And for replication errors, the pathway used is called mismatch repair. So for base excision, the damaged base is detected by an enzyme, and then DNA glycosylase comes in and removes the base, and then an endonuclease comes in and cuts out the remainder of that nucleotide. Then DNA polymerase comes in to add a new nucleotide in that place, and DNA ligase seals it together. In nucleotide excision repair, the DNA is open to form a bubble, and then the damaged DNA is excised by a nuclease. And then DNA polymerase comes in and replaces the DNA that has just been removed, and then DNA ligase comes in and anneals the fragments of DNA. And in mismatch repair, again, the defect is the mismatch is cut out first by an endonuclease, which makes which cleaves the DNA at one site, and then an exonuclease comes in and removes the nucleotides until the mismatch is removed. Then a DNA polymerase comes in and replaces the sequence that was just removed, and DNA ligase then anneals the DNA fragments together. And then for double-strand breaks, there are two methods to repair this type of DNA damage. Both can be used for the same purpose. Homologous recombination or homology-directed recombination is more accurate, but it requires the presence of a template strand, which may or may not be necessary depending on where in the cell cycle the cell is. So for homology-directed recombination to occur, the cell needs to have a replicated copy of DNA. So it has to be either after after DNA replication has completed, but before the replicated copies of DNA have separated. So while both can compete for repair of a double-strand break, non-homologous end joining is more likely to occur because it doesn't have the necessity of a template strand. So what happens in non-homologous end joining is that the DNA ends of the double-strand break are recognized by an enzyme, and then a complex known as the non-homologous end joining complex assembles at the double-strand breaks, or DSB, and the ends are then bridged to promote stability and process so that they're able to ligate. Then the broken ends are ligated to result in a repaired double strand of DNA. Whereas in homologous recombination, the 5' ended strand is resected to create a 3' overhang of in the broken DNA. Then the 3' end of the broken DNA then invades one strand of invades homologous DNA double strand, and the invasive strand will displace one strand of the homologous chromosome to create a complex known as synapses. So then DNA replication occurs to create an identical copy of DNA, and then the synapses is resolved and the two double strands are separated. So compared to non-homologous end joining, a homologous recombination is not as error prone, but it requires the presence of the second homologous chromosome. And there are consequences to DNA damage and the response to DNA damage. So among these are inhibition of cell cycle progression. So this can include inhibition of transcription, replication, and chromosome segregation, which can result in apoptosis, which is programmed cell death. It can also set off these mechanisms known as cell cycle checkpoints that can arrest the cell at various points in the cell cycle. When this happens, the cell can will either be arrested until the DNA damage is resolved, or it will either undergo apoptosis or senescence, where it'll die. the other option is DNA repair. If DNA repair occurs without any errors, the cell will survive. However, if the repair is error prone, this can result in mutations or chromosomal aberrations, which can result in cancer, aging, or a disease state. Okay, and that's all I have for DNA damage and repair. Are there any questions? Okay, so I think
Yeah, let's stop the recording so that we can have a little bit more like free space. Yeah. Okay.
okay, so now we're going to get into DNA techniques. Okay, so the first one I'm going to talk about is DNA purification. So this is a pretty simple concept. So you can extract DNA from multiple sources of cells. The first thing that needs to happen is cell lysis, which is just breaking apart the cell. This can usually involve some kind of detergent or some kind of mechanical breakdown of the cell, depending on what type of cell it is. If it's a eukaryotic cell without with just a cell membrane, just detergent may be sufficient. But if it's bacterial or fungal or plant with a type of cell with a cell wall, there may be need for some kind of mechanical destruction as well. Then using the cell lysate, generally undergo multiple wash steps to remove the membrane lipids, to break down the proteins with a protease, and then remove RNA using an RNase, which breaks down the RNA. And then the DNA is then either eluted through a filter column, or it's separated from the rest of the cell components using base separation. So this can be done through multiple techniques. There are commercial kits that can do this. In addition, many labs have in-house methods of doing this. So I know Beth has a really good protocol for DNA extraction from strawberries. I was wondering, could you talk about that a little?
Yeah, I love things like this. Yeah. Yeah, so it really is, strawberries are great because they have 8 times the number of chromosomes that human cells do, or I guess 4 times, whatever, a lot. They have a lot of DNA. So yeah, the process is basically the same parallel process that you do in a lab, just with like household ingredients. So instead of like lysing them with like special chemicals, you're using soap, dish soap, salt, and water, and you're using physical crushing lysis methods with your hand just by smushing it in a strawberry or in a plastic bag. And then to remove the lipids and other sorts of like wash steps, basically you just filter it through a coffee filter. And so because the DNA is so small, it can pass right through that filter. And again, because you're doing kind of a crude prep, you don't really need it to be super pure from all of those proteins and lipids and things. But then you just add alcohol, and the alcohol will make the DNA kind of stick and clump together, and it will come out of the liquid solution, out of that phase. So it really is a 5-minute process, and it's really fun to mirror what you do in the lab with what you can do at home.
Yeah, I have a similar technique that I use in the lab. So for E. coli DNA extraction, normally I use a commercial kit, but for, most of my work is actually with yeast, and to get genomic DNA out of yeast, what I do is I use glass beads to mechanically break apart the cell wall. And from there, I take the cell lysate and I mix it with chloroform and an aqueous solution, some kind of a salt-based buffer. And then I just spin it down. All the protein and lipids go to the bottom with the chloroform, and all the DNA is in the aqueous solution on top. So that contains DNA and RNA. So then I take out the aqueous layer, add RNase to it, treat it for however long it takes the RNA to break down, then use ethanol to salt out the DNA, and just wash out anything that's left. And the whole process takes like 30 minutes without any kind of fancy equipment. Okay, so next I'm going to talk about some applications for purified DNA. So one that I think a lot of people are already familiar with is PCR, or polymerase chain reaction. This is essentially a form of in vitro DNA replication, where you're able to make multiple copies of DNA using variations in temperature and added enzymes and nucleotides. So there are a list of reagents that are necessary for this to for a PCR reaction to be successful. If any of them are missing, you're not going to get DNA replication. So the first thing you're going to need is a template DNA. This can be either purified DNA or a plasmid, like I mentioned before, that you already have constructed, and that'll act as your template. You're going to need individual deoxynucleotides. So those are your A's, T's, G's, and C's. You're going to need a DNA polymerase, a very specific kind that I'm going to get into next. You're going to need magnesium ions. A lot of enzymes require a cofactor. In the case of DNA polymerase, magnesium ions act as the cofactor. So in vivo, when DNA is being replicated in the cell, you need primase to create those short RNA primers. You're not going to have that in the test tube, so you need to create your own primers that are basically short sequences of DNA, and you have to add those in to your reaction. And lastly, you're going to need a salt buffer to keep the pH at the appropriate pH. So the type of polymerase you need for PCR is it has to be one from one that's thermostable. So the reason for this is because you don't have helicase. The way you're going to separate the double strands of DNA is by applying heat to it. So you need a polymerase that's going to be functional after being exposed to high temperatures. A really common polymerase that's used for this is called Taq polymerase. That's short for Thermophilus aquaticus. It was isolated from Thermus aquaticus. It was isolated from bacteria that grows in hot springs, and it's able to withstand high temperatures. It's a critical enzyme that's necessary for PCR reactions, and it, like most polymerases, it works in the 5' to 3' direction. Because as I mentioned before, when you're working with PCR, you need to include primers that are specifically designed for your region of interest of DNA. So what you do is you pick two regions that flank the part of the segment of DNA that you're interested in, and for PCR, you need both a forward primer and a reverse primer. So the forward primer will be a copy of the forward strand. So when you're looking over your the DNA sequence, you'll be looking at the the sequence of the forward strand. The forward primer is approximately 20. Is a region that's approximately 20 base pairs, and it should be upstream of the DNA sequence that you're interested in. Now, the reverse primer is going to be going in the opposite direction. So what you do then is you take your DNA sequence and you get the reverse complement of it. And then from there, you construct your reverse primer, which will be a short reverse complement of the forward strand. And these two primers should ideally be flanking either side of your DNA region of interest. And when you separate, when the two strands of DNA are separated after applying heat, the primers will then anneal to the appropriate strands, and that is where DNA polymerase can start producing the new DNA strands from. So there are 3 steps of PCR. The first one is denaturation. This occurs at a very high temperature, usually about 95 degrees Celsius. And the purpose here is to separate the 2 strands of DNA. Second step is the annealing step. At this point, the primers are going to anneal to the 2 template strands. This in this example, it's set to 54 degrees Celsius. It can vary depending on your primer. So as I mentioned last week, the number of guanine and cytosine bases you have will affect the temperature at which a strand of DNA will melt. So the more G's and C's you have, the higher the melting temperature will be. Usually when you're deciding on an annealing temperature, it's about 5 degrees Celsius lower than your lowest primer melting temperature. And the last step is extension. So here it's shown as 72 degrees Celsius. This varies depending on which polymerase you're using. In the case of Taq polymerase, 72 degrees Celsius is the appropriate temperature for catalytic function of Taq polymerase. After the first cycle of PCR, you're going to have templates that are annealed to template strands annealed to new DNA. And then in the second cycle, you'll have the templates separating from the new DNA, and then more new strands being formed using the original template and the new DNA as templates. So by the third cycle, you'll have primers binding to the amplicons that were produced in the previous cycle. These 3 cycles are repeated multiple times. Usually it's between 25 and 30 times, and each time the number of DNA amplicons you have will increase twofold, and by the end of 30 cycles of this, your ultimate yield will be 2 to the 30th copies of your DNA of interest. So PCR has had massive impact to science, particularly in genetic research, gene mapping, cloning, DNA sequencing, and gene detection. It's also been used heavily in medical tests and treatments. I'm sure you're all familiar with this now because a form of PCR called RT-PCR or qPCR were in use for COVID testing. So something that's very relevant today. It's also present in a lot of popular media and movies and TV as well. So any questions about PCR? Sorry, my cat is like harassing me right now. Yeah, any questions?
You mentioned qPCR. Can you say what that is?
So that's quantitative PCR, where you're monitoring the amount of the number of amplicons as they're being produced.
So say again, vary the amount of amplicons? Like,
yes. So it's a way of monitoring how many copies of a gene you have in a template. As the amplicons are being formed, the thermocyclers are monitoring how much of the gene is, or how many copies are being made.
oh,
It's a way of telling like relative abundance, like how much of your RNA sample or whatever is that particular gene, or like for really small amounts of things, it's really good at detecting them like at very low amounts, whereas like you might not see it if you just waited until the end. So that, you know, some of the applications of it are for like high sensitivity testing. Yeah.
So you end up getting like a measurement of how much DNA was originally present.
Yeah.
So after you've done the amplification, you do some further processing on it that measures how much was produced, and then it works backwards.
It's done during the process,
Yeah.
which is pretty amazing.
Oh.
The machine
Yeah.
Wow.
measures like fluorescence while it's copying.
Mhm.
Yeah.
Oh, I see.
It's also useful in certain research applications where if you're trying to like introduce a mutated version of a gene into an organism and you see like a massive effect, and you may not know if it's because of the mutation in the gene or if it's because you just put in a ton of that, you have put in multiple copies of the mutated gene or not. Actually, one of my colleagues had a paper retracted because of that. Not retract. I think he just had to do like a massive revision on it because it turned out the massive effect on the in vivo function of this gene was actually because he was putting in like 8 copies of a mutated gene as opposed to 1. Yes, so it's useful. Okay, so any more questions? Okay, yeah, so I'm going to talk next about visualization of DNA by gel electrophoresis. So this is just a basic overview of how, that's, what are you doing? Sorry, my cat. Okay, okay, so here what you do is you take your sample of DNA, you mix it with a dye and load it onto an agarose gel. The agarose gel is then placed in an apparatus where electrical current is applied, and your DNA travels from the cathode to the anode because of the, and this is because of the DNA being negatively charged. And the agarose matrix then allows for the DNA molecules to be separated by size. So DNA is transparent, so you need a way of visualizing it. So there are 2 dyes involved in gel electrophoresis. One is the loading dye, and this is just a color dye that's used to visually monitor where the DNA is on the gel as it's running. The other dye is called lithium bromide. There's also an alternative called CyberSafe because the lithium bromide is a mutagen. Basically what lithium bromide does is it binds the DNA and it fluoresces under UV light. So that will allow you to see the specific fragments of DNA under UV light. So one thing to note here is that the loading dye won't show you exactly where each fragment of DNA is. It'll just show you, it'll just give you a sense of where the smallest fragments of DNA are on the gel. And again, because DNA is negatively charged due to the phosphates in the backbone, it's able to run from the negatively charged cathode at the top, at the front end of the gel to the positively charged anode at the bottom of the gel. And because of the agarose matrix, the smaller fragments of DNA will pass through the pores in the agarose faster than the larger molecules of DNA, and that will separate the DNA by size. And in order to determine the size of your DNA, you also run what's called a marker or a ladder, and this contains fragments of known size, and this will give you an indication of what size your DNA fragments are. Okay, so any questions about electrophoresis?
The first set of dyes, how do you get different colors?
Yeah, so there's a few different types. So these are commercially made. So some of them are they're just plain blue. Some of them separate as they run down the gels, and they'll separate based on size. I'm not really sure how they do it because these are commercially made.
Oh, I see.
Yeah.
You just like mix it in, and then when you run the gel, you get these different colors.
Yeah.
Yeah, the companies have mixed it, but we can do it. Like we can just add together like 3 or 4 different food colorings, plop that in a gel, and it will separate them by size. So it's the same process. Yeah. And I see.
What does one dye connect up with the larger fragments or the smaller fragments that it separates out by color?
Sorry, I don't think I understood your question.
Probably I don't understand what it's doing.
It's just separating by size. So the pigments of the food dye are different size. So it's just,
the pigments of the food dyes are
yeah, they're just different sizes. So
so you put those in, and
yeah.
that's like a ladder.
Yeah.
Because you just see the different colors and where they I thought that somehow the DNA was
Nope. Nothing.
that's got to be with the ethidium bromide and the fluorescence and everything.
Yeah, that's right.
Okay, thanks.
Yeah, I see that Lucy and Thomas both have questions in the chat.
Okay.
Yeah,
the ethylene bromide then reacts with the DNA, so you don't have the pure DNA at that point. Is that correct?
so like if you're trying to purify it out of the gel, Thomas, is that what you're asking?
Yes.
Yeah. So
Well,
you want to, yeah, you go, Shirley.
Yeah, it's doable. You can extract DNA from an ethidium bromide containing gel, but you have to put it through another process called gel purification or gel extraction. It's pretty commonly done, but usually you use a commercially made kit for that.
And really often what you're using the gel for is just to see like, did your reaction work or did X and Y and Z happen? So you don't actually need that DNA back. You just want to know, and you don't use your whole sample usually.
Well, I recall, I think that we wound up cutting the gel in order to isolate that segment of the DNA. And it would seem to me that if you had the ethylene bromide in there, you'd have contaminated the DNA. It wouldn't be.
Yeah, and that's exactly what Shirley was just talking about. You put it through another kit.
Right, purification.
Yep, exactly. And then the ethidium will wash.
You got to remove it. Yeah.
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 Okay. Just wanted to clarify.
Yeah, that actually is something that is pretty commonly done. It's especially during ligations, which I'm going to talk about in a bit. And sometimes when you do PCR, you have multiple bands, which can happen if your primers aren't specific to, if there's more than one site in the genome where a primer can attach to. Sometimes you get multiple PCR bands or bands resulting from PCR, in which case you would cut out the correct one and purify it from there. Just some examples of when gel purification would be useful. Okay, so I'm going to go into molecular cloning now.
Hold on, Shirley. There's another question from Lucy.
Okay.
Does the sequence have any impact on how fast the DNA goes through a gel?
I think it's just size that would make that would control that, as far as I know. Yeah.
That's my understanding too. There may be some very subtle differences, like if it's super GC rich, it might move a little differently, but I don't think it has a significant effect, and definitely not anything that you could visualize.
Okay, I'm going to go into molecular cloning now. You don't need to pay attention to the details in this figure yet, but I'm just going to give you a quick overview first. So molecular cloning basically involves the use of plasmids, which we've talked a little bit about before. So what you can do with plasmids in a laboratory setting is you can take a gene of interest and add it to a plasmid to create this vector, and then you can transform it into another organism such as E. coli or yeast, and then select for the cells that contain this plasmid through a process called transformation, and then using these cells, you can generate recombinant proteins or generate more of more of this plasmid with your gene of interest. So just want to go over what plasmids are. So plasmids are distinct from bacterial DNA. So bacterial DNA are a long continuous chromosomes which contain all the genes and regulatory information for the entire organism. Plasmids, on the other hand, are while they're also made of DNA, they're short, circular, and they're extrachromosomal, meaning they're separate from the chromosomes, and they only contain a few genes. So again, they're circular DNA molecules. They're self-replicating and separate from the chromosome, meaning they're not non-genomic. They do occur naturally in bacteria, and they can be found in the environment. Usually carries one or more genes. It can carry foreign genes, as well as genes with specific functions, and they're propagated by environmental uptake, which includes transformation, horizontal gene transfer, or cell division. So plasmids can be isolated from bacterial cells through the same process that we talked about before. You just lyse the cell walls, separate the cellular debris from the DNA, and then separate chromosomal DNA from plasmids, because they're generally distinctly different sizes. This could be done just by centrifugation usually. So plasmids.
How do you separate the plasmids from the chromosomal DNA? Did you say by? I'm sorry. Go ahead.
Again, there's usually done by commercial kits, and there's a step in the lysis process where that's time sensitive. If you let that go for too long, you end up getting chromosomal DNA in your sample. But if you keep it to a specific time point, then you only get your plasmid DNA. And you can confirm this just by running it on electrophoresis. Usually with a plasmid, you know what size you're looking for, whereas with chromosomal DNA, it's going to look more like a smear, or it's going to be a different size.
Mm-hmm. Yep. Thank you.
Okay, so one of the techniques that's used to manipulate plasmids to insert a gene of interest is restriction enzyme digest. This is a process that uses enzymes that cleave DNA molecules at specific sites that are usually palindromes. These enzymes are called restriction enzymes or restriction endonucleases. So they target DNA at specific sites and cleave them to leave either sticky ends or blunt ends. So sticky ends are what's shown here. So there's a 5' overhang on both of the resulting fragments of DNA. In a blunt end, you wouldn't have this overhang. It would just be blunt here. And this is just an example of a plasmid called the P-glow plasmid, and I just want to point out the components here. So this plasmid has an origin of replication and an ampicillin marker, so ampicillin resistance marker. So what this gene does is it gives the bacteria that contains this plasmid resistance to ampicillin. It also has GFP, which is the gene of interest in the case of this plasmid, as well as RFC, which is, in this case, a transcriptional regulator of this particular plasmid. But what I want to highlight here is that this plasmid also has known sites for various restriction enzymes. So here you can see that there's one cut site for ECHO R1, there's 2 for HIN3, and 3 cut sites for BAMH1. So because we already know where these sites are, we should be able to predict how many DNA fragments you're going to get when you cut the plasmid with any combination of these enzymes, and what sizes the fragments should be. So in the case of this enzyme, if you cut with HIN3, which has 2 cut sites, you end up with 2 fragments. With BAMH1, which has 3 cut sites, you end up with 3 fragments, and with ECHO R1, there's 1 cut site, so what you end up with is 1 linear piece of DNA. So this is what you would expect for circular DNA. If you started out with a linear piece of DNA, you would have different results. For instance, if the linear piece of DNA had 2 cut sites, you would end up with 3 fragments, not 2. So just something to keep in mind when you are predicting predicting what fragments you're going to get when you plan a restriction enzyme digest. So this brings me back to the overview for molecular cloning. So here you have a hypothetical plasmid where containing a multiple cloning, a site where you can insert your gene of interest. So you would treat this this plasmid as a vector. So you would perform restriction enzyme digest to open up the plasmid to create a space for your gene of interest to be inserted, and then you would treat it with a phosphatase to remove any phosphate groups. What this will do is it'll prevent this plasmid from closing in on itself. And then you would purify this plasmid as a single piece of linear DNA. At the same time, you would prepare your insert. So this can be done by PCR, or it can be cut out of another plasmid by restriction enzyme digest. Ideally, what you would have is an overhang on either side of your vector, as well as your insert that are complementary to each other. If you don't have that, what you could do is use PCR to add in a restriction enzyme site to your insert. Next, you would take your treated vector and your insert and undergo a ligation. So this will insert your gene of interest into the vector using an enzyme called DNA ligase. Some things to keep in mind when this is done is you have to consider the vector to insert ratio. There should be an excess of the insert compared to the vector. Reaction time and temperature can also be dependent on multiple factors, including what type of ligase you're using and your quantities of vector and insert. And the ligation step will give you a completed plasmid that contains your gene of interest as well as some kind of selectable marker. This is then inserted into an organism such as E. coli or yeast. In this case, they're showing E. coli. This is a process known as transformation, and you would confirm that that your vector has been taken up by E. coli by some kind of a screening method. So for instance, if your vector contained an ampicillin resistance marker, if your cells have been successfully transformed with your vector, they should be able to grow on ampicillin. Whereas if they didn't take up your vector, take up the plasmid, then those cells should die on an ampicillin containing media. And then from there, you would take your transformed cells and confirm by PCR or sequencing. And if you're working with a recombinant protein, you can then culture those cells to generate more of your protein of interest from E. coli, or sometimes this can also be done in yeast. So any questions about cloning? I know that was a lot.
Yeah, I'll echo. A lot of this stuff is stuff that's like, you just have to start doing it. You know, you just have to start doing it and it will make more sense, right? Right. I don't know about you, Shoily. What was it like when you were first learning it? Were you like, okay, I kind of understand this, but like until
Oh,
you
that was,
with it.
I
Yeah.
think that was like 15 years ago. I can't remember that far back.
Yeah. Jeremy, you want to put the recording on pause again so we can have a little more open dialogue?
Sure.